and it lets the it lets it expand.
Yeah.
It's kind of like stacked and it slides, and then it allows it to contract.
In the kidneys is similar.
I'm sorry?
In the kidneys is what I was talking about. I've been reading about the kidneys recently.
Oh yeah.
About the cuboidal cells
Mhm.
and the absorption.
Yeah, because cuboidal cells are associated with
Absorption?
absorption, secretion are with the cuboidal cells. So like columnar cells are like usually your mucus cells.
Mm-hmm.
They secrete mucus almost every time, but they don't absorb anything. Why? They're too tall.
I was thinking that maybe that was gonna be a stupid answer.
They're too tall.
They're too long.
There's just, I mean, they're just too tall
Yeah.
for, you know, stuff that gets absorbed into it wouldn't make it out the other end.
Right.
But cuboidal and simple, or not simple, cuboidal and the word that will come to me later, squamous, thank you, are thin enough that things can just go boop right there. Let's see. This test is a lot of like multiple choice things that are difficult to ask. And it's a lot about filtering. The renal corpuscle consists of the glomerulus and the glomerular capsule. True or false? Say that again.
True.
True. The corpuscle is the glomerulus and the capsule.
Okay.
Yeah. So it's both of them. So you have the glomerulus and then the capsule. If you add them together, you have a corpuscle.
Okay.
Which I also think is a kind of gross word. They're kind of the two grossest systems, just kind of lumped together. It's kind of gross. Let's see. Well, so when you have in the glomerulus, when you filtered the blood and the liquid that you now have in the proximal convoluted tubule, what is that liquid called? Filtrate. Yep. It's not urine yet. When does it become urine?
In the collecting duct?
The collecting duct, yep. Because can you change the concentration at all in the collecting duct? Can you change anything about the urine?
I don't think so.
Nope. Once it is in the collecting duct, it's done. You're done changing it. So the distal convoluted tubule is your last chance. Yep, pretty much. And so after it comes out of there, then it's urine. And it's just gonna go. Let's see. You would typically expect glucose to be filtered but not reabsorbed. And this is kind of just a concept question since we haven't really gone into it, but you'd filter, when you filter stuff first, right? When you filter stuff in glomerulus, you filter all of it. You get it all out. You just take it all out because you're not sure what's good or what's bad, right? Mm-hmm. And then in the process, you select what's good and what's bad, right? So if you filtered all of the glucose, filtered a bunch of glucose out, right? Would you want to reabsorb that glucose or would you want it to stay in the urine?
Does it depend on glucose levels?
Not really. Generally, you want to absorb glucose. You don't want glucose in your urine because glucose is pure energy.
Right.
Like, you don't have to do anything to that. You just send it off to the mitochondria and it's pure energy. So you want to take that glucose in. So what disease has trouble reabsorbing glucose?
Diabetes mellitus.
Diabetes, yeah. So if you have diabetes, it means sweet urine, which is really gross because it will, there's enough sugar in there that if you were to taste it, which I think they used to do to test diabetes, yes, so nasty, but it'd be sweet.
Wow.
because there's just so much glucose in there because their blood sugar is so high. Their body's just like, God, like we can't reabsorb this.
Right.
So you know there's a problem when you have glucose in your urine.
Why can't the sugar that's coming out of you, why can't like it be reabsorbed into the body with diabetes?
Well, if you have so much sugar in your bloodstream, your body's just gonna get overwhelmed. It's gonna be like, we just can't, like, we need to get some of this out.
Because
some of the sugar out.
from the
Yeah, but obviously filtration, like, it doesn't solve the problem because you don't have the proper hormones, you don't have the insulin working right to regulate it. So it
that's
just,
why it has to come out, right? Or
it's just because there's so much in the blood, there's just so much in the blood that you just can't reabsorb all of it. Your body just won't reabsorb all of it. But it means that there's a problem because you don't want any glucose in your blood or in your urine.
Right.
And do you want protein in your urine?
I mean,
No. No.
Protein stays behind, right? Or can't be.
If you have
filtered out
an injury, then you get protein in your urine. Sometimes, also if you're pregnant, you can get protein in your urine. Like sometimes it's fine, but usually if you have protein in your urine, it means something gone wrong because you're not supposed to have protein.
how can you tell? Like, is it?
Did you guys do the urinalysis? There's a test you can do. Someone pees into a cup and then they dip a stick into it with a couple different tests, a couple different like little areas on it, and then those areas turn different colors and they look on the bottom and compare it, and it tells them like what the concentration of protein is, the concentration of glucose is, and a bunch of other stuff. Let's see. So in the proximal convoluted tubule, you find lots of large mitochondria. Why? So what are you doing?
most reabsorption happens in the proximal.
Exactly. So you're pumping things. So you need lots of energy, right? So you have lots of mitochondria because you're actively pumping things and you're using a lot of ATP. Yep. Basically, anywhere where you're pumping stuff, you're going to find a lot of mitochondria. So if a question like that comes up, it's like, why do you have a lot of mitochondria here? Just think about what it's doing. And if you're pumping stuff, it's because you need a lot of energy. Let's see. Trying to find ones that we've gone over. Yeah, this one. Presence of proteins in the urine could indicate damage to the filtration membranes. So, kind of a little. This will help a little bit. So, what ion do you think is most important for urine, like concentration? So urine it's not sweet. What is urine generally?
Sodium.
Yeah, it's sodium. It's salty. So sodium is going to play a huge part. And basically, what's going to happen is in the loop of Henle specifically, the body is going to selectively pump sodium in and out of the membrane, and it's going to let the water follow it. So it's going to determine how much water it wants to keep in the blood and how much water it can leak out in the urine, right? And it will pump the sodium in or out of the loop of Henle depending on what it wants. So that's how your urine gets concentrated. It gets concentrated in the loop of Henle by pumping sodium in and out, sodium and water. So the most important nutrient for reabsorption is sodium, because if you pump sodium, what's going to happen with water? It's going to follow it. Yep. So the entire time you're talking about the nephron, if you move sodium, water follows it. Let's see.
It's so cold in here.
I know, honestly, my hands are so cold.
Freezing.
I can't feel my feet. I mean, maybe that's because I'm sitting on my feet and I'm probably killing the nerves.
No, it's not. I'm like frozen.
Let's see. All right, most of these we haven't gone over. Well, here you go. So if urine output is greater than fluid intake, blood volume will go up or go down.
Urine output is greater than fluid intake.
Mhm.
Down.
It will decrease, yep. And what will happen to blood pressure?
It will decrease.
Decrease, yep. So that's why the kidneys are so intimately involved in blood pressure,
Right.
because the more, if you're not drinking enough, that's why your urine becomes more concentrated, because the body wants to keep all of that liquid in you so that your blood pressure stays up. Yep. Alrighty. Yeah, so that was that, and it's 11:58. So both your timing. Any questions? Or anything?
Not really.
No.
Did
Okay.
you send out the practice test email like already, or are you gonna do it?
I'm gonna do it.
Okay.
I'm gonna do it right now, actually.
So I'll take this over tonight and then.
Yeah, and then I'll just be here. I'll just be here at 2. Yeah, if you want to come, you can come. If you don't want to, you don't have to. You don't.
Okay.
I will not be offended if you stay home and enjoy your first day of winter break.
Yeah.
I was not expecting it to be this cold today.
I know. I didn't either. It does have the answers, but not on it.
Oh.
I mean, I didn't print it out though.
Okay. Cool. So there's like an answer key. that
There
you can take
is.
Without knowing the answers.
Yes.
Okay, perfect. I was gonna go on and do the mastering, low quiz test things.
Mhm.
I was gonna do those tonight.
Nice.
Just to kind of see what I know.
Yeah. And on this practice test, there's a couple of things that it goes into more detail about stuff than we need, but it's helpful. So a gallstone that blocked the common bile duct would cause fat content in the feces to increase, decrease, or stay the same.
Nope.
So if the common bile duct was blocked, would increase. Why would it increase?
The bile wouldn't be breaking down.
Exactly. So what does the bile do?
Emulsifies fat.
Emulsifies fat, yeah. So how does it allow the fat to be broken down?
It just breaks it down into
It breaks the fat into little droplets. It separates the fat into little droplets and it increases the surface area. So it allows the enzymes more room to digest it. Yeah, so he specifically mentioned it increases the surface area, which is why I mentioned it. Norepinephrine would cause secretion of stomach acid to increase, decrease, or stay the same. Bless you.
Can you say it again?
Norepinephrine.
Thank you.
Would it increase, decrease, or cause the stomach acid to stay the same?
That's blood, right?
Mhm.
So it would be decrease.
Decrease, yep, because norepinephrine is
Inhibits.
sympathetic. Yep, exactly. Let's see. If plasma levels of secretin and cholecystokinin, did I pronounce that right? Yes, cholecystokinin increased, the volume of bile in the gallbladder would blank. And the pH of the duodenum would blank. So we'll work through that. So first of all, what is cholecystokinin? Where does it come from?
I don't know where it comes from. All I know is when I wrote it down.
So cholecystokinin, it comes from the, or I think it comes from, well, it doesn't say where it comes from, but it influences the pancreas and the gallbladder. So cysto means gallbladder.
Okay.
So cyst is is the medical abbreviation for gallbladder.
Okay.
So it increases the pancreatic enzymes, and it increases production and secretion of bile, and it slows stomach emptying. So basically, once the food gets there, the cholecystokinin causes the pancreas to dump enzymes, and it causes the gallbladder to dump bile, and it shuts off the sphincter, and it allows just that little amount to be digested right there. So you don't want to add anything to it, you want to keep that there for a minute. So if our cholecystokinin levels increased, what would happen to the volume of the gallbladder? Decrease, because it's causing the gallbladder to empty, right? So what would happen to the pH of the duodenum? Would it go up or down or stay the same? It would go up, right? Because what are the secretions of the pancreas? Are they acidic or basic?
Acidic.
They're basic. They're basic to counteract the acidity of the stomach. Because only in the stomach do you want that high acidity. As soon as it gets into the duodenum, you want to cut that off. You want to neutralize it, or else it'll burn the duodenum to shreds, and you will have no duodenum.
That would be sad.
It would be sad.
Would you say pancreatic enzymes, or what is it?
Cholecystic. Basic pancreatic enzymes
Are basic.
are basic, yeah. Basically, if it's being secreted in the duodenum, it's basic.
Okay, I see.
Which is where, like, if you're not quite sure what the answer is, stomach is always, you want more acidity in the stomach, but you want less in the intestines. So it's a pretty fair guess that because I can't think of anything off the top of my head that's acidic in the intestines in general.
Right.
So if it's being secreted in the intestines, it's basic. So what cells secrete pepsinogen? Chief cells. Yep. Hello. Yeah, chief cells secrete pepsinogen. And what does pepsinogen do?
Breaks down protein.
Breaks down proteins. But first, before pepsinogen breaks down proteins, what has to happen to it?
It needs to turn into pepsin.
It needs to turn into pepsin. What turns it into pepsin?
Hydrochloric acid.
Hydrochloric acid. Exactly. Let's see.
What is intrinsic factor?
Intrinsic factor.
So it says parietal cells secrete hydrochloric acid and intrinsic factor. Sheep cells secrete pepsinogen. What is intrinsic factor?
It ups B12 absorption, and I'm pretty sure it influences red blood cell production. I'm gonna look up a different definition of it. Intrinsic factor. Oh, I have no internet again. This internet is stupid.
I'm sorry, pepsin breaks down
Protein.
protein.
Alright, I have no Yeah.
Huh.
I don't know. Remind me at the end, and I'll work on getting getting this back up. Alright, let's see. We're just doing like a little, I found a practice test. I'm gonna send it out in an email, but we're going through it and we're just figuring out what we know about the digestive system.
Okay.
Let's see. That one's too hard to say out loud. During defecation, the external anal sphincter will lengthen and the rectal muscularis will. So, which one will relax? Which one will contract?
Well, the external sphincter will relax.
Mm-hmm. Yeah, the muscularis will contract. Exactly.
What was it? The muscularis?
Yeah, rectal muscularis.
Hmm.
Which, I mean, even if you didn't really know that it existed, it's pretty easy to tell where it is.
Right.
Let's see. All of the following are components of saliva except, so there's pepsin, IgA antibodies, mucin, and salivary amylase. Which one is pepsin? Exactly. Because where does protein digestion happen? In the stomach. Only the stomach, right? So what does amylase break down?
Starch.
Starch and sugar, yep, because starch and sugar. And what does lipase digest? Fat. Yep, exactly. Let me see. Some of these are too hard to say out loud. So where does chemical digestion of carbohydrates begin? In the mouth. Yep. Which is kind of gross. If you chew a cracker for long enough, it will digest in your mouth. I mean, you have to chew it for a long, long time, but I've not done it personally because that would be just nasty, but it could happen. You could chew a cracker long enough to digest it in your mouth. And I think that'd be really, really gross. We don't know that one. Let's see. So where do so following digestion and absorption, most dietary fat content can be found within, so what do they go, in the intestines, right? They get absorbed. What vessels do they get absorbed into? Yeah.
Like blood vessels? I don't understand.
Okay, so blood vessels or lymphatic vessels?
Oh, the lymphatic.
Lymphatic. And what are those little lymphatic vessels called? Lacteals.
Oh, right.
Yeah. Yep.
Yes.
Yeah, so intestinal lacteals. True or false? A function of the ileocecal valve is to prevent backflow from the large intestine to the small intestine.
False. It's reverse. Yes.
True.
Say it again.
The ileocecal
Yes.
valve.
Prevents.
Backflow from the large intestine to the small intestine.
Oh, that's true.
Mm-hmm.
That makes sense.
True or false? A lack of functioning salivary amylase would prevent an individual from being able to digest lactose.
False.
It is false, yep. There are separate enzymes for digesting lactose. It's lactase. And a lot of the enzymes are named for what they digest. So like lactase, sucrase, digest sucrose, right? Let's see. Waves of muscular contractions that propel the contents of the digestive tract from one point to another are called
Peristalsis.
peristalsis. Yep. So where do you find the secretory cells, like the chief cells and the parietal cells in the stomach?
In the gastric pits.
In the gastric pits. Yep.
It just sounds so gross.
I know, it really does. Like, honestly, the digestive system is just really nasty. What's the purpose of having villi and microvilli in the intestines?
The brush border.
Not so much the brush border. There's a simpler.
To push, to move things.
They don't move things. Peristalsis moves stuff.
Mucus, they produce mucus.
To absorb and stuff. Nutrients. Absorption.
But why would you have so many little projections?
For a higher surface area.
Higher surface area. Yep. Higher surface area means that there's more contact area for food to touch the brush border and for things to get absorbed into. So it means you can pack a lot of surface basically into a really small area. Otherwise our intestines would be huge, or they'd be really inefficient, and we'd have to eat all the time, and it'd be nasty. Let's see. The portion of the small intestine that attaches to the large intestine is the ileum. Yep. An intestinal hormone that stimulates the pancreas to release a watery secretion that is high in bicarbonate ion is called. Say that again. It's an intestinal hormone that stimulates the pancreas to release a secretion that's high in bicarbonate. It's very alkaline.
Is it what we just talked about? The cholecytokinin?
It's not. It's actually secretin. So secretin causes the pancreas to secrete.
Okay.
Yeah.
So anything that the pancreas secretes is going to be a buffer or going to be basic.
Basically, yeah, because you're right in the duodenum, right, and you just have all of this extremely acidic stuff. Like think car battery acid, acidic level. Like I'm not even kidding.
No.
This stuff is, I know it's crazy, but this stuff will it will like digest your finger. If you were to stick your finger in stomach acid, your finger's gone. It can, I think they've done studies where they've like stuck pencils in stomach acid and just pencil's gone.
The whole pencil's gone.
Yeah, like you can just like dip it in there and then you have no more pencil on that end wherever it's stomach acid touch. Like this stuff is serious, so you want to get as much alkaline stuff in there, as many bicarbonate ions in there as you can before it eats the duodenum. Let's see. Okay, an intestinal hormone that stimulates the gallbladder to release bile is cholecystokinin. Yep, and I don't know if y'all were here. Cysto in the digestive system means gallbladder.
Say again.
Cysto. When you're talking about the digestive system, if you see something with cyst or cysto, it's talking about the gallbladder. So cholecystokinin means that it causes the gallbladder to release stuff.
Okay.
It's one of the functions. But like if you ever see someone has cystitis, means their gallbladder's inflamed. Yeah.
The cystis? Is that what you said?
Cystitis.
Cystitis.
Mhm.
Okay. Okay.
Yeah.
Gotcha.
So we normally think of a cyst as like a little fluid-filled sac,
Right.
but I mean it's kind of what the gallbladder resembles.
I have a question.
Mhm.
If, if anything with the word cyst has to do with the gallbladder, what about the cysts that, women get in their, like on their ovaries and things like that?
It's not everything.
Right.
It's a medical term that you have to use, like you have to have context for.
Mm-hmm.
So like if we're talking about the digestive system
Mm-hmm.
and it has cyst in it, it's most likely referring to the gallbladder, but it's not necessarily true
Okay.
elsewhere in the body.
System.
Yeah.
Oh, okay.
Yeah, it does. What is the vein in the liver lobule called in the center of it? Nope, it's actually just a central vein. But it's the one, it's right in the middle of the liver lobule. and it's just called the central vein. It's too easy, but it's there. Let's see. What's the main function of the large intestine? Water? Yeah, water absorption. Exactly. How much of the water gets absorbed?
10%?
10%. Yep. Decreased levels of bile salts in the bile would interfere with the digestion of fat. Yeah. Bile, anything bile is digesting is fat. Bile digests fat. Which is why if you get your gallbladder removed, I knew someone whose dad got their gallbladder removed, and he like had to cut fat and like basically anything with, you know, any level of fat, he had to just cut it out because he couldn't digest it very well. All right, let's see. We'll do a little bit of the urinary system after this one. Let's see. So, does anyone remember what the order of the histology of the tissues? There's like layers of the tissues in the digestive system, right? There's 4 layers. Anyone remember what the layers are?
Mucosa, submucosa.
Mm-hmm.
I don't remember the other one.
Muscularis externa, and then
Starts with an S, right?
serosa. Yep. Yeah. So mucosa, which makes sense. We're secreting mucus in the lumen, right? And then submucosa, which is right next to mucosa. Muscularis externa, because there's a lot of muscles involved in the digestive system, and then the serosa on the outside. Yep. Let's see. As the food in the stomach is repeatedly squeezed and mixed with gastric juice, it is converted into a creamy paste called chyme, yeah, which is really nasty.
A creamy paste.
Yeah, honestly. Let's see. So here's a question that encompasses a couple systems. So which of the following is not considered an endocrine organ? The stomach, the colon, the duodenum, or the pancreas?
Pancreas is both an exocrine and an endocrine.
Mm-hmm.
Okay.
So
What's
basically,
only an exocrine?
huh?
What's only an exocrine?
Which one's only exocrine?
Was that the question?
No, which one is not considered an endocrine? Sorry.
Oh, it's not an endocrine.
Mm-hmm. So basically, out of here, which one doesn't secrete, one of these doesn't secrete anything?
Stomach?
The stomach secretes a bunch of stuff. The colon.
Oh,
The colon doesn't secrete anything.
Right.
So regardless of whether it's endocrine or exocrine, it doesn't secrete anything. It just absorbs stuff.
Right.
Let's see. A lot of these are like multiple choice that would be hard to read out, so I highly suggest like looking through this in the email because it's got a lot of good questions in here about the liver and liver lobules and stuff. What's in a portal triad? Where do you find it? So portal system,
Yeah,
liver, right? So we're talking now about liver lobules, right? Little hexagon shapes. So what's a portal triad? There's 3 things in there.
Did we talk about this in lecture? Because I'm not.
We kind of briefly went through it, but basically a portal triad is just 2 blood vessels and 1 bile duct.
Okay.
Yeah, because you're filtering the blood and then you're making bile. So a portal triad is 2 blood vessels and 1 bile duct.
Okay. It's on
Yep.
the model.
Yeah, we kind of like skimmed over it in lecture though. We just kind of were like, here's a liver lobule, and now it's gone.
Yeah.
So that's why I brought that one up. Let's see. Where do you find Tinea coli? Large or small intestine? Actually large. What is the Tinea coli? Yeah, exactly. What is the Tinea coli? It's that band of muscle that runs along the colon, and it causes the bunches, right? What are the bunches called?
Yeah.
Starts with an H.
Hofstra.
Hofstra. Yep. Yeah, and that little band of muscle helps with peristalsis because there's very little muscle in the actual wall of the large intestine. So you've got this band of muscle that kind of helps propel everything through.
It's a smooth muscle.
I believe, yeah.
Obviously.
Yeah. All right, let's go. I'm gonna grab the one for the urinary system. We'll go through, if I can, yeah. We'll go through the parts of the urinary system we did go through in lecture. And then I guess we're gonna finish it today. And then we'll be done.
I know I don't like. This week is the last week of class, like Friday technically. Yes.
Mm-hmm.
Why is he trying to rush through it and not have the exam due like on Thursday? You know what I mean?
Yeah,
Like,
I
is he
don't.
trying to rush and get it all in by tomorrow? You know, we could have had one more lecture. We could
Yeah,
have
I
slowed
don't-
down a little bit. You know.
I don't really know. I mean, he hasn't mentioned anything to me, so
Maybe he has deadlines.
yeah.
But we don't know.
He might, I mean, he might have meetings scheduled on Wednesday that are mandatory that he can't get out of
Yeah. Right.
because he's part of like all the boards.
It's okay. Tomorrow night. Oh, I know. When I saw that, I looked at it like on Saturday. I'm like, what?
Yeah.
And when is due?
No, like it's due tomorrow night.
Tomorrow night?
Yeah, it's like the deadline.
At like 11:30, I think. Mhm.
I know.
Yeah. Which is why I was wondering if anyone would want to come instead of, because we won't have SI Wednesday because there'd be no point. If anyone would want to come at 2 p.m. tomorrow for the urinary system. So I'm gonna be open to that. You don't have to give me an answer now, but I'm gonna send out an email. I'm gonna send out an email today.
I'll be there.
Okay. I'll send out an email today and I'll probably just do it anyways. So at 2 p.m. we'll be here, or if this portable's not open, we'll find a portable. We'll find somewhere. So that we can go over the urinary system and answer any questions for, take tests. Alright, let's see. What are podocytes? Where are they found?
In the glomerulus.
In the glomerulus. What do they do?
They help filtrate. Or they surround the capillaries.
Yeah, they kind of like, think of pod, like feet, right? And they kind of like wrap around the blood vessels and the capillaries in the glomerulus.
Do they have any other function than just wrapping around? Or that
They
we
like
need to know?
add the filtration. They add to like the slits and stuff. Like there's slits in the blood
yeah.
vessels, and then they kind of add more
Yeah.
filtration function.
So what is osmotic pressure, first of all? What does that mean?
I mean, it's
Pressure due to what?
solute concentration.
Solute concentration. Yeah. So it's pressure due to osmosis. So remember, so what is the difference between osmosis and just diffusion? Thank you. I was like filtration. It's not good. So diffusion is just solutes. You know, you have a sugar cube, right, in the glass, and you don't want all that sugar in one area, so it just dissolves and it spreads throughout the glass. But what is osmosis? What's the difference with osmosis?
The water moves towards the greater solute concentration.
Exactly. So osmotic pressure means that the water is going to follow the high solute concentrations. So basically, if you pump out a bunch of solutes and you filter out a bunch of solutes, like out of the blood, the water's going to follow it. So that's what osmotic pressure is talking about. If you move solutes, water follows. Let's see. What is the does anyone know what the cells in the stomach, what kind of cells are in the bladder, I'm sorry, in the bladder, the epithelium